A Punnett square is the way that we can see where genes arrange themselves in the offspring. In this example, we will take a single set of genes and see how they arrange in the offspring. First, there is some terminology that we need to establish. A dominant characteristic is one that only needs one copy of an allele in order for the offspring to have that particular characteristic. There, the characteristic dominates. A dominant characteristic is represented by a capital letter. A recessive characteristic is one that needs two copies of an allele in order for that characteristic to be shown in the offspring. Blue eyes is a good example of this. A person with blue eyes must have two copies of the blue eye gene, while brown eyes is dominant, and a person needs only one copy of the brown eye gene in order to have brown eyes. The recessive characteristic is represented by a lowercase letter. Codominant is where both copies of different alleles are expressed in the offspring. For example, if you have a plant which has genes that can provide for both blue and white flowers, and the plant has both of these, then the plant will have white flowers with blue speckles or blue flowers with white speckles. Incomplete dominance is where these two characteristics are blended in the offspring. For example, if you have both blue and white genes in the same plant and these are incompletely dominant, then the flowers will be pastel blue in color. Homozygous refers to the condition where both of the alleles in an offspring are the same. This could be two dominant or two recessive. Heterozygous refers to the condition where both of the alleles in an offspring are different. For example, cap A, small a. Genotype is the genetic makeup of all the alleles that are contained in the individual. The phenotype is the physical expression of all the genes in the individual. Remember that the physical expression is often based on other triggers, so that you may have a gene, but it may not be expressed. Now, let's look at the scenario of a single gene Punnett square. First, our example will use the gene for height. In this case, we will call capital T tall and lowercase t short. Our parents in this case are going to be heterozygous. That is, they have one copy of each gene. Through meiosis, the gametes are formed with each gamete, in this case, having either a capital T or a lowercase t. We place each possible into the Punnett square. Yes, it looks like a tic-tac-toe board in the upper row or the left column. We then add the letters, remembering to place any capital letter first and then any lowercase letter. This gives us 4 possibles. When we look at the genotype by counting how many of each we have, let's look. There is 1 cap T, 2 cap T lowercase t, and 1 lowercase t. When we look at the phenotype through the genotype, now first let's remember that a T is dominant, tall, so we count up how many of the genotypes have cap T. There are 1 cap T and 2 cap T lowercase t, which gives us 3 tall individuals. Then we count how many of the genotypes have lowercase t, and there is only 1, so we have 1 short individual. Let's check the math. 3 plus 1 equals 4, which is correct. So our phenotypic ratio is 3 tall and 1 short. With a Punnett square, we can determine how the genetics of different genes get passed on.